Hi everyone, this is Glenda Ganzon and welcome to my Human Anatomy and Physiology class. And for today's lesson, I'm going to discuss the cell growth and cell division. So stay tuned. In this video, you will be able to learn the importance and prevalence of cell division. Well, there are a few cells in the body that do not undergo cell division, such as gametes, red blood cells, most neurons, and some muscle cells. Most somatic cells divide regularly. So, a somatic cell is a general term for the body cell, and all human cells except for cells that produce eggs and sperm, which are referred to as germ cells, are somatic cells. So what is somatic cells? Somatic cells contain 2 copies of each of their chromosomes, 1 copy received from each parent. A homologous pair of chromosomes is the 2 copies of a single chromosome found in each somatic cell. So the human is a diploid organism, meaning having 23 homologous pairs of chromosomes in each of the somatic cells. The condition of having pairs of chromosomes is known as diploidy. So cells in the body replace themselves over a lifetime of a person. For example, the cells lining in the gastrointestinal tract must be frequently replaced when constantly worn off by the movement of food through the gut. But what triggers a cell to divide, and how does it prepare for a complete cell division? The cell cycle is a sequence of events in life of the cell from the moment it is created at the end of the previous cycle of cell division until it then divides itself, generating 2 new cells. This is a picture of the cell cycle, and the 2 major phases of the cell cycle include mitosis, which is the cell division, and interphase. So when the cycle grows and performs all of its normal functions, interphase is further subdivided into G1, S, and G2 phases. So one turn or cycle of the cell cycles consists of 2 general phases. First is the interphase, followed by mitosis and the cytokinesis. So the first one is the interphase. What is interphase? It is the period of the cell cycle during which the cell is not dividing. And the majority of the cells are in interphase, and most of the time. Mitosis is the division of genetic material during which the cell nucleus breaks down and 2 new fully functional nuclei are formed. Cytokinesis also divides the cytoplasm into 2 distinctive cells. In the interphase, a cell grows and carries out all normal metabolic functions and processes in a period called G1 or the gap1 phase. The gap1 phase is the first gap or growth phase in the cell cycle. For the cells that will divide again, G1 is allowed by replication of the DNA during the S1 phase or the synthesis phase. So the synthesis phase is the period during which the cell replicates its DNA. And after the synthesis phase, the cell proceeds through the G2 phase. So the G2 phase is the second gap phase during which the cells continues to grow and makes the necessary preparation for mitosis. And between the G1, S, and G2 phases, cells will vary the most of or in their duration of the G1 phase. And it is here that a cell might be spent a lot of hours or many days. And the S phase typically lasts between 8 to 10 hours, and the G2 phase approximately 5 hours. So in contrast to these phases, the G0 phase is a resting phase of the cycle or the cell cycles. And cells that have temporarily stopped dividing and are resting, this is a common condition, and cells that have permanently ceased dividing, like the nerve cell, are said to be in G0 phase. This is a picture of a homologous pair of chromosomes with their attached sister chromatids. The red and the blue colors correspond to the homologous pair of chromosomes. and each member of the pair was separately inherited from one parent. So each chromosome in the homologous pair is also bound to an identical sister chromatid, which is produced by DNA replication and the result in the familiar X shape. So billions of cells in the human body divide every day, and during the synthesis phase of 2 interphase, the amount of DNA within the cell precisely doubles. Therefore, after DNA replication, but before cell division, each cell actually contains 2 copies of each chromosome. So each copy of the chromosome is referred to as a sister chromatid, and it's physically bound to the other copy. The centromere is the structure that attaches one sister chromatid to another, and because a human cell has 46 chromosomes, and during this phase there are 92 chromatids in the cell, make sure not to confuse the concept of a pair of chromatids, one chromosomes and its exact copy attached during mitosis, and a homologous pair of chromosomes, which is 2 paired chromosomes which were inherited separately from each parent. The mitotic phase of the cell typically takes between 1 and 2 hours, and during this phase, the cell undergoes 2 major processes. First, it completes mitosis, and during which the contents of the nucleus are equitably pulled apart and distributed between its 2 halves. Cytokinesis then occurs, dividing the cytoplasm and the cell body into new cells. So mitosis is divided into 4 major stages that take place after interphase. And in the following order, number 1 is Prophase, second is Metaphase, the third is Anaphase, and the fourth one is the Telophase. The process is then followed by cytokinesis. So the first phase of mitosis is what we call as the prophase, during which the loosely packed chromatin falls and condenses into visible chromosomes. During prophase, each chromosome becomes visible with its identical partner attached, forming the familiar X shape of sister chromatids. The nucleus disappears early during this phase, and the nuclear envelope also disintegrates. So a major occurrence during the prophase concerns a very important structure that contains the origin site of the microtubule growth. Let's recall that the cellular structure holds centrioles that serve as origin points from which microtubules extend, and these tiny structures also play a very important role during mitosis. So a centrosome is a pair of centrioles together. The cell contains 2 centrosomes side by side, which begin to move apart during the prophase. And as the centrosomes migrate to 2 different sides of the cell, microtubules begin to extend from each like long fingers from 2 hands extending toward each other. And the mitotic spindle is a structure composed of the centrosomes and their emerging microtubules. So near the end of the prophase, there is an invasion of the nuclear area by microtubules from the mitotic spindle, and the nuclear membrane has disintegrated, and the microtubules attach themselves to the centrosomes that adjoin pairs of sister chromatids. The kinetochore is a protein structure on the centromere that is the point of attachment between the mitotic spindle and the sister chromatids. So in this stage is referred to as the late prophase of the pro-metaphase to indicate the transition between prophase and the metaphase. The second stage of the mitosis is what we call as the metaphase, and during this stage, the sister chromatids with their attached microtubules line up along the linear plane in the middle of the cell. So a metaphase plate forms between the centrosomes that are now located in either end of the cell. The metaphase plate is the name for the plane through the center of the spindle on which the sister chromatids are positioned. So the microtubules are now poised to pull apart the sister chromatids and bring one from each pair to each side of the cell. Anaphase is the third stage of mitosis. Anaphase takes place over a few minutes when the pairs of sister chromatids are separated from one another, forming individual chromosomes once again. So these chromosomes are pulled to opposite ends of the cell by their kinetochores. As the microtubules shorten, each end of the cell receives one partner from each pair of sister chromatids, ensuring that the two new daughter cells will contain identical genetic material. So the last stage of mitosis is what we call as the telophase. In telophase, it is characterized by a formation of two new daughter nuclei at either end of the dividing cell. And this newly formed nuclei surrounds the genetic material, which uncoils such that the chromosomes return to loosely packed chromatin. Nuclei also reappear within the new nuclei and the mitotic spindle breaks apart, each new cell receiving its own complement of DNA, organelles, membranes, and centrioles. And at this point, the cell is already beginning to split in half as cytokinesis begins. In cytokinesis, the cleavage furrow is a contractile band made up of microfilaments that forms around the midline of the cell during the cytokinesis. This contractile band squeezes the two cells apart until they finally separate. So the two new cells are now formed. One of these cells enters its own cell cycle, able to grow and divide again at some future time. And the other cell transforms into the functional cell of the tissue, typically replacing an old cell there. So imagine a cell that completed mitosis but never underwent cytokinesis. In some cases, a cell may divide its genetic material and grow in size but fail to undergo cytokinesis. So this results in larger cell with more than one nucleus, and usually this is an unwanted aberration and can be a sign of cancerous cells. So as the cell proceeds through its cycle, each phase involves certain processes that must be completed before the cell should advance to the next phase. A checkpoint is a point in the cell cycle at which the cycle can be signaled to move forward or stop. At each of these checkpoints, different varieties of molecules provide the stop or go signals depending on a certain condition within the cell. A cyclin is one of the primary classes of cell cycle control molecules. Let me introduce to you what is a cyclin-dependent kinase, or what we call as the CDK. A CDK is one of a group of molecules that work together with cyclins to determine progression past cell checkpoints. And by interacting with many additional molecules, these triggers push the cell cycle forward unless prevented from doing so by stop signals. If for some reason the cell is not ready at the G1 checkpoint, the cell must be ready for DNA synthesis to occur. And at the G2 checkpoint, the cell must be fully prepared for mitosis. And even during mitosis, a crucial stop-and-go checkpoint in metaphase ensures that the cell is fully prepared to complete cell division. And the metaphase checkpoint ensures that all sister chromatids are properly attached to their respective microtubules and lined up at the metaphase plate before the signal is given to separate them during anaphase. May I discuss to you the implications when the cell cycle is out of control? Most people understand that the cancer or tumors are caused by abnormal cells that multiply continuously. If the abnormal cells continue to divide unstopped, they can damage the tissues around them, spread to other parts of the body, and eventually result in death. So in healthy cells, the tight regulation mechanism of the cell cycle prevent this from happening, while failures of the cell cycle control can cause unwanted and excessive cell division. Failure of control may be caused by inherited genetic abnormalities that compromise the function of certain stop and go signals. So environmental insult that damages DNA can also cause dysfunction in those signals. And often, a combination of both genetic predisposition and environmental factors leads to cancer. The process of a cell escaping its normal control system and becoming cancerous may actually happen throughout the body quite frequently. And fortunately, certain cells of the immune system are capable of recognizing cells that have become cancerous and destroying them. However, in certain cases, the cancerous cells remain undetected and continue to proliferate. If the resulting tumors does not pose a threat to surrounding tissues, it is said to be a benign and can usually be easily removed. If capable of damage, the tumor is considered malignant and the patient is diagnosed with cancer. So this ends my discussion about the cell growth and the cell division and also mechanism of the cell cycle, particularly with the implication if the cell cycle is out of control. And if you have any question, please write it in the comment section down below and I'll be glad to answer them all. And once again, I would like to thank you all for watching this video and for listening to my discussion. Until my next video, bye everyone!